What I would like to do is to focus on the issue of neural tube defects, as this is one of the most important clinical manifestations of abnormal development of the neural tube. So I would like to present this story, Neural Tube Defects, Reviewing Embryonic Development with an eye towards future interventions. I want to remind everybody that neural tube defects are clinically significant. They result in lifelong disability. The nerves that innervate the bladder, the bowel, sexual function can easily be compromised in infants with these disorders. Secondary issues from hydrocephalus lead to learning and developmental problems, and of course the obvious orthopedic problems come to mind. There is a strong correlation between the level of the lesion and the degree of disability. We see a much poorer survival rate in those children who have neural tube closure defects above the 11th thoracic vertebra. We have a much better survival rate for those with lower defects. And over the years, we've come to appreciate that some, but not all, neural tube defects are responsive to folic acid. So just a reminder, the neural tube is one of the earliest systems to develop in the embryo. It forms initially as a neural plate. This neural plate is many cell layers thick. There will be an elevation of the neural folds by supporting underlying mesodermal tissue, so that this once neural epithelial plate is now folding towards the midline with areas of hinge point bending, the dorsolateral hinge points. And ultimately, if everything goes well, these hinges of the neural folds will meet and fuse on the dorsal aspect of the embryo and complete a closed neural tube. This neural tube will give rise ultimately to the brain and spinal cord. However, if failure to close completely happens in the rostral end of the embryo, you end up with a lethal condition we call anencephaly, whereas closure below the level of the 12th thoracic vertebrae is referred to as a meningomyelocele or spina bifida. Of neural tube defects, we see on the top part of this slide rostral defects. So we have cranioracheschisis, where the neural folds fail to elevate at all, anencephaly. We see a post-neurulation closure defect called encephaloceles, and then we have iniencephaly, which has a rostral, a stargazing appearance where there's usually one or two encephaloceles. On the bottom part of the slide, you'll see a range of posterior defects, spina bifida occulta, which may be a closed neural tube defect, only recognizable by perhaps a tuft of hair on the skin, closed spinal dysraphisms or lipomas, meningoceles, where we have an extrusion of the meninges and cerebrospinal fluid, but not neural tissue, and then an open spinal cord defect within the meningeal cyst, which we call a myelomeningocele or spina bifida. Consider neural tube defects to have a prevalence rate now in the United States between 0.5 and 0.8 per thousand live births, and that works out to about 1,500 infants born every year with spina bifida and another 850 cases of anencephaly. But globally, the story is quite varied. We have, by best estimate, about 500,000 neural tube defects births annually. Given their terminations, that number is hard to figure out. What is important is that there are various parts of the world where the rates of neural tube defects can be quite high, including China, which we believe to have over 130,000 neural tube defects every year. India also has nearly that many. Consider neural tube defects to be a complex trait, a complex genetic disorder. There are strong genetic evidence, even though less than 10% of neural tube defects are syndromic. We see twin concordance data where like-sex twins are more often both affected or both unaffected, and we see the various prevalence amongst different ethnicities, as I just mentioned. but there is no clear Mendelian inheritance. A couple with one affected child only has a 5% risk of having another. If you have two or more affected children in a family, the increased risk is only now 10%. So it's clearly non-Mendelian. We know from mouse studies there are over 400 gene mutations that give you a neural tube closure defect phenotype. So it's unlikely that any one gene is going to explain a significant portion of the population burden of neural tube defects. Thus, we like to look at neural tube defects as a result of having a genetic and environmental interaction. And thus, despite decades of study, we really are far away from understanding the causes of most infants who are born with a neural tube defect. Great modifier of the neural tube defect risk, and that's folic acid or vitamin B. Folic acid is an essential vitamin. The body cannot make it, create it. It has to be consumed. In vivo folic acid, the synthetic form, is reduced to a bioactive tetrahydrofolate. It enters the cell as a monoglutamate, and it's polygglutamated to remain within the cell cytoplasm, where it has an amazing range of functions. It's one of the physiologically most important compounds around. It's involved in the synthesis of nucleic acids, amino acids, neurotransmitters, and methylation reactions. We know that folic acid being important, we have now, since 1998, have fortified the American food supply with folate, and as a result, it has increased the population levels of folic acid, so that prior to food fortification, the 50th percentile was about 12 nanomoles per liter. Now, about half the population has 35 or more nanomoles per liter of folic acid. Had an impact on neural tube prevalence rates. No matter how you administer the folic acid, whether you give a multivitamin with 400 micrograms of folic acid a day, whether you fortify the food supply, wherever it's been tried, in North China, Newfoundland, New England, Hungary, on and on down the list, you reduce the background prevalence of neural tube defects to about 1 per 1,000. So as a successful impact on reducing the prevalence of neural tube defects, and as a result, we are only able to protect or prevent about 15% of the global population of neural tube defect births. Folic acid is important in modifying the risk. Many geneticists and scientists and clinicians thought that somewhere in this folate pathway, this one-carbon metabolism pathway, lies the secret to understanding susceptibility to neural tube defects. Rather than study the 35 to 50 different enzymes that are involved in one-carbon metabolism, my group focused on the folate transport mechanisms, as there were only 3 particular gene families that transport folates into cells: the folate receptors, the reduced folate carriers, and the proton-coupled folate transporters. The expression pattern of FOLR1 in the mouse, which is equivalent to the human folate receptor alpha, or FR1, you can see the blue staining representing the expression of this gene, and in the earliest embryos you can see expression in the mid-cervical region where neural tube closure is thought to begin. It progresses bidirectionally, and you can see the intense blue staining all the way down in the last frame to the posterior neuropore. So you can see the expression of the folate receptor alpha gene. It's expressed at the right time during neural tube closure. It's expressed in the right place within the neural tube, and it has the right job, which is to regulate intracellular folate concentrations. The older embryos, you can see the intense staining in the midbrain and closing up the face. In editing techniques and knock out this gene, you can reproduce the whole range of neural tube defects that we see in humans in the mouse. So This served as validation that folic acid and folate transport genes are really important in determining susceptibility of like looking for a needle in the haystack. Now, we typically considered complex birth defects like neural tube defects in this multifactorial threshold model, so that an individual embryo would inherit a genetic liability which would put him closer to the threshold of abnormal development or further away. Well, it would take less of an environmental insult to move that embryo that is genetically susceptible over the threshold and express a neural tube defect. We considered this to be multifactorial threshold inheritance. But as sophisticated, more recently we are aware that a more fitting model would be an omnigenic model of complex phenotypes. In this model, we can assume that most traits can be directly affected by a modest number of genes or pathways that have a specific role in the disease etiology, and we call these core genes. And these core genes have largely interpretable roles in disease, such as genes involved in one-carbon metabolism. And when these core genes have loss of function mutations, that's when they have their strongest effect on disease. Well, given that genes operate in networks, and that most genes are really only a few steps away from the nearest core genes, so that really all of the genes that are expressed at a given time during embryonic development will have some minor role in the heritability of that defect, much more so than the core genes themselves. We looked at the distribution of singleton loss of function variants in neural tube defect cases in 3 different cohorts: in a Chinese cohort, in a US population cohort, and in a cohort from the Middle East. And we found in each case the number of singleton loss of function variants are greater, significantly greater, in NTD embryos than those from unaffected controls. So you can see in the Chinese cohort they had 15 loss of function variants, whereas the controls had 6, and similarly in the US and Middle Eastern cohorts. So if you assume by that reasoning that the more singleton loss of function variants an individual has, the greater the risk they are for having a neural tube defect, then if you consider an anterior defect like anencephaly, which is lethal, to be more severe, anencephaly embryos to have more loss of function variants than spina bifida cases. And you can see that this is true, the red bar being anencephalic, the blue bar being spina bifida, and we see a greater number, nearly 15 loss of function variants in anencephalics relative to the 9 in spina bifida. So calculate the number, the odds ratio of neural tube defects with different numbers of these singleton loss of function variants. And as you can see, when the number of singleton loss of function variants reaches 9, the odds ratio for having a neural tube defect is now greater than 1. When an individual carrying more singleton loss of function variants than 9, the risk for neural tube defects increases exponentially. So at 10, there's a 4.8-fold increase. At 12, it's about 6.5-fold increase. And by 14 singleton loss of function variants, there's a 43-time greater incidence of neural tube defects. When to map them to different KEGG pathways, we could see that the incidence of loss of function variants fell into many different GO categories, including metabolic pathways, olfactory pathways, PI3K, AKT signaling pathways, tight junctions. They were all over the place. So the impact of any one gene is contributing to the risk of neural tube defects in this omnigenic model. Just an idea about treatment. We've been looking for genes. We know lots of different pathways, but now, and we know that there are many, many different. So one approach that we've been using is to use a tool called an androgenetic haploid stem cell, which is basically a haploid stem cell from a male mouse that is basically a sperm without tails. We can use CRISPR-Cas9 technology to insert or humanize these cells with known combinations of human neural tube defect variants and insert these into oocytes and create mice that have neural tube defects. We've also patient-derived induced pluripotent stem cells from which we could then have a steady source of material to replicate, provide them with growth factors or neurotrophic factors, and find out which way to activate a healthy matrix to perhaps close the defects. For this, we have various different mouse models that we can use, one of which that is particularly helpful is the FKPP8 knockout mouse. This mouse gives 100% penetrant spina bifida lesion, which you can see in this figure. And these animals are live-born. They can live multiple weeks, dragging themselves with their functional forelimbs to their mothers to nurse. We also have a sheep herd that has a spontaneous 15% prevalence of neural tube defects, spina bifida, in the lambs. We can use matrices that we develop in the mice. We can then ultrasound use and determine which ones are carrying pregnant, which one are carrying embryos that have spina bifida, and do in utero treatments so we can demonstrate the effectiveness of these treatments. In closing, I'd like to just reiterate that the genomic architecture of neural tube defects is complex. It remains to be a challenge to discern the various sensitive approaches to DNA-based prenatal diagnostic procedures, but it is a work in progress. We know that periconceptional folic acid reduces the risk of select malformations, including neural tube defects, and it's still incumbent upon us to figure out how this works. And finally, the elucidation of the genetic pathways often compromised in neural tube closure defects represent important tools to aid in the development of effective intervention and treatment programs. I'd like to acknowledge the generous support we've had from the National Institutes of Child Health and Development. Thank you.